Hello and welcome to this internet webcast, Applying Milk Fat Globule Membrane and Human Milk Science to Improve Nutrition for Infants and Toddlers, facilitated by the Annenberg Center for Health Sciences. As mentioned, this certified webcast provides continuing education credits for physicians, pediatric nurse practitioners, nurses, and registered dietitians. A link to submit for CE credit will be provided after the presentation. It is now my pleasure to introduce our speaker, Rafael Jimenez Flores. Dr. Jimenez Flores is the J.T. Parker Endowed Chair in Dairy Foods at Ohio State University. He earned his Ph.D. at UC Davis in Food Science and Agricultural Chemistry. In this webcast, Dr. Jimenez Flores describes how dairy processing advances have isolated and purified milk fat globule membrane from cow's milk for supplementation to infant formula. He also reviews the structure and composition of milk fat globule membrane and its biological activity, as well as the evidence for cognitive development and immune system support. Dr. Jimenez Flores will answer questions during the last several minutes at the end of this activity. Dr. Jimenez Flores, you may begin your presentation, starting with our learning objectives.
Thank you very much, Victoria, and thank you very much to all the organizers, and good day to all the audience, present and I guess future. So as I always start defining the learning objectives to my students, and in this case my audience, the first learning objective is going to describe how dairy processing has succeeded in isolating this fraction from milk, and how this is important for supplementation to infant formula. And we're going to see how the bioactivity is reflected once the component is added to the infant formula. So first of all, let's find out how we find ourselves in the research world where we are and what those advances we have. So according to a survey on the dairy processing, we have several underutilized dairy components such as lactose, the minerals that are in milk, but more important for the talk today is that the milk fat and phospholipids is an area of very active research and a resurgence in the terms of importance in nutrition. So we really need to know more as scientists from the biology and biochemistry about the milk fat globule membrane. Where does it come from, and what is its structure? So just as a background on the social media and what is out there, there may be still some remnants of maligning the fats from milk or animal origin. The vast majority of our research now, and supported by many studies, really have shown that it's not fair to demonize specifically dairy fat. And that has opened the gates for really renewed information, renewed research on this area. And you may, as an audience, be asking, well, what does the milk fat globule membrane have to do with everything nutrition and especially infant nutrition? Some of the questions from my students and the audiences that I talk to is milk fat globule membrane similar in human and cow's milk? What are the contribution of the phospholipids to its structure? Is that structure of any importance? How do we obtain this ingredient? And how about inclusion in the formula? So we'll take it by parts, and let's start by the components. I would say they're not the major nutritional components like protein, water, sugars, but it definitely in the milk fraction component of the milk fat globule, we have components that are very important, and they have been studied in many other fields on brain function, like is choline, sphingomyelin, gangliosides that were initially isolated from ganglia, cholesterol, sialic acid, and some other very specific lipids that are glycosylated lipids called cerebrosides. And all of these are found in the milk fat globule membrane. On the immune defense system, we have mucins, butyrophilin, lactoferrin, something very interesting like toll-like receptors, which are indicators and signals about pathogens in our diet. So all of this is condensed in what I consider to be one of the more interesting frontiers in dairy science, and especially milk chemistry. What you should be looking at right now is an electron micrograph of a fat globule being secreted into the milk lumen by a mouse milk secretor cell. These cells are classified as epithelial cells. They're all in the mammary glands of all mammals. Anything that produces milk has these cells, and they're very well conserved, as we will see later on. And you will see that big gray sphere is composed almost exclusively of triglycerides. So that's what would be the butter fat. And you will notice it is surrounded, it's perfectly on the margin of a very thin line. That thin line is the membrane, which at that point is a 3-layer, because if you look at the part that is touching the cell on the bottom of that sphere, you'll notice a very thin line, and that's the initial monolayer of phospholipids that then pushes through and gets secreted, or as my students in food science would say, it's microencapsulated into the milk. If the next picture shows us with even more luxury of detail how thin this membrane is, and hence why it is so challenging to study it, because it really, although we can see the effects as we pour cream in our coffee and it doesn't make us leak the way that it would if we added, let's say, mayonnaise or something that is not encapsulated by a membrane like that, we can see that the dimensions are exquisitely small. You can see by comparison, we cannot even look at it under normal microscope, the casein micelles, and yet that membrane is even smaller. So let's explore what happens at the biological point of view on secretion of milk. I apologize if in the audience are already milk secretion experts. This cartoon depicts with the endonucleus, the Golgi apparatus right in front of it, where a lot of activity gets done to synthesize proteins, as we can see. And then that E is a vesicle, it's a secretory vesicle. But on your left-hand side, you'll see how the microlipid droplets grow from very minuscule droplets that they keep aggregating, like the drops of fat on your chicken broth, until they reach to a larger size. And by the time that they get to the apical membrane, which you can see where the upper part is where we have the milk, and inside the cell, that fat needs to be extruded through the membrane, which engulfs it perfectly, and then we have our little droplets of cream in the milk. I always put these post-secretional changes with a question mark, because it's my belief that Mother Nature really didn't intend milk to be bottled or put into a piece of cheese or anything like that. And it is up to us food technologists to know and to mimic whatever happens with that membrane later on. Now, this process has been long studied and is more complex than what I'm describing, because there's many other pathways of secretory vesicles that we're just continue to understand. And as you see in the electron micrograph on your right-hand side, the lipid droplets is really surrounded by a lot of the other smaller vesicles that are present in the milk of all mammals. And that is been very recently reviewed and beautifully explained by Dr. Ian Mathers and his team. by using a technique that is, if you ever have a chance to see this paper, I really recommend, is an intravital imaging. And this is done with a small rodent, a rat, in which the live rat is surgically intervened to expose the mammary gland onto the confocal microscope. And this is very important because the mouse, as you can see on your top right, is kept alive. That is a blanket to keep the mouse as comfortable as it is with an exposed mammary gland to the microscope. And then the microscopist, it takes a bunch of pictures in three-dimensional. So first I want to show you the cartoon of the alveoli, of the milk secretion alveoli on your left-hand side, and see the similarity in your panels J and K on these fluorescent pictures. The red dots are fat globules that are inside the cell. In your panel M, you can see, and this is the newest contribution by this group, that the fat globules are there ready to go, but they're not secreted into the lumen until there is a very specific signal, or oxytocin. Once oxytocin is injected to the mouse that is comfortably laying on top of the microscope, there is a rain, literally a rain of lipid droplets that get secreted into the milk. So hopefully at one point we'll have this in a movie, or you can go and see that paper by Masendunskas, and it is fantastic as a added information. You have that. Just so that we emphasize this point, this is a very, very well-conserved mechanism across the mammalian world. And what it is very impressive is the amount of fat globules that we have, especially early lactation. We have smaller globules. If you were to add all the surface of those little spheres, you could, with a glass of milk that has all of these globules in there, cover a surface that is similar to a tennis court. So that's what physical chemist tells me. I've done some calculations, and my goodness, it is incredible what happens, and that would be on normal milk. But there's something else that is very important to this discussion, at least to the biological part of this discussion, and that is that back there in 2008, my friends Bionas and Juan Lor, Maximum Bionas, published the whole metabolic pathway of all the enzymes that are needed to secrete the fat to milk. In this case, it goes upside down to the other pictures because the milk is at the bottom of the slide, and you can see where the fat little globules get zipped by the membrane. But the important part about this slide is that it gives us, for the first time, a whole list of enzymes that we could see and compare between humans, cows, goats, donkeys, horses. And it was amazing to see that those enzymes that are involved in this very process that you're looking are 99% conserved, which means that the gene ontology indicate that these are some of the most conserved genes in metabolic pathways, which to any biologist or understanding of life tells that this is a process that is very important according to nature because, it hasn't been modified. All right, now that we have seen a little bit about the milk fat globule membrane and where it comes from, and I'm hoping that my audience is starting to have a new appreciation for what we have in milk, and not just that we ask for a non-fat milk in our coffee with foam or without foam. We are going to show to you some of the industrial processing steps that we have to isolate this particular component. We can isolate it from cream, from butter or buttermilk, from whey, and our main focus is to keep in check the thermal treatments that we do to our milk, because heat, as we know, changes the conformation of proteins and the protein and lipid interaction. So here is a diagram of the modern butter churn. When I say butter churn, most of the people that read history think of a nice wooden bucket with a stick that a dairy maiden is shaking. This is far from that. What you see is this butter churn gets fed the cream on the top. Then it has a very fast churner that is designed precisely to break the milk fat globule membrane, and thus the triglycerides get separated from the serum, as you see by the screw mechanism in which we squeeze out the serum that we call buttermilk. Coincidentally, buttermilk, even though the name has butter on it, has less fat than your regular skim milk. And then, of course, butter has 40% fat. And in this nation, we produce vast amounts of buttermilk. But it's not the buttermilk you buy in the supermarket. That is just fermented skim milk. Right now, the bulk of our buttermilk goes to big dairy plants, and it gets dried into powder. And if you ask me where is it used mostly, it's used mostly in bakery and ice cream, where the properties of the phospholipids are great as emulsifiers. So where it all starts? Well, if you're a researcher like me, or you want to start getting your milk fat globule membrane for your infant formula, you identify a very good producing dairy farm, you get the milk. The milk then is processed through a machine that is called a cream separator, because it gives us the skim milk, as is shown here by the blue line on the bottom, and the cream. Now, a well-known trick of the dairy processors is that we separate skim and cream, and then we remix it so that we can give you 1% fat milk, 2%, 3%, or as is whole milk. When I talk of how much fat is there in a whole milk, it sounds like a lot of fat. It's only 3.5% fat. We could sell a product that is 96.5% fat-free, but dairy farmers decide not to go that way. And right there in the glass, I'm trying to depict that that's where we get the cream. And that cream is a raw material for making butter. I'm going to exemplify how we get milk fat globule membrane from butter. The main reason, and in this picture you'll see me here lecturing my students here at the Ohio State University. Those that take dairy processing get to work with an automation machine that says EGLI right there. That's a continuous butter churn. And as long as you feed cream to this machine, it gives you butter. You can see it in the bucket right there, the butter. Or you see the red arrow that is pointed to the left points to a tank, where that tank was added for our research, and it collects all the buttermilk. How does the buttermilk look? Well, just like skim milk. Right there on your right-hand side in the bottom panel, you see the appearance of real buttermilk. Well, like any industry, what we do, we can research it as such, liquid. Hopefully, we'll get some advances, and I'm told that in Europe, in Ireland, they're starting to have drinks directly, like it used to be 50 years ago, of real buttermilk. Maybe somebody will commercialize it. For the moment, what we do industrially is pass that buttermilk after some concentration through a spray dryer. Here's an example of a pilot plant spray dryer. The ones in industry are 4 or 5 stories tall. This is not even one, it's about 10, 12 feet tall. And we can get powders. And those powders are of very interesting characteristic when they have a milk fat globule. The next slide, this slide shows us an electron transmission electron micrograph. On your left-hand side is just skim milk. treated with different heat treatments, but that's just how the electron microscope looks at the casein micelles that are in skim milk. So there's no cream there, no fat. And the buttermilk, although I told you that it has no fat, it does have those phospholipids in a very peculiar form. If you notice on your right-hand side picture, there's a lot of wormy-like looking there, like little serpentine figures. And that happens to be the original very thin membrane, but is rolled over, like if you were to roll over a carpet where your kids have been playing with their Legos and their toys and everything. So the carpet happens to be the membrane, and the Legos and the toys are the proteins that are inherited to the milk from the mother. So that's why we think that there's something very interesting about these little wormy things, because we do believe, and we have seen some evidence, that these are means of communicating nutritional signals to the baby. And in case of the industrial buttermilk, they're all encased in this component. Well, that's all fine and dandy, but how are we going to separate that for our use, say, in infant formula? As you can see, this is not a normal thing that would happen in regular processing. And since we industrialize our milk, this is not a normal part of our diet. So we use an advance called ultrafiltration. And so whether this can be done from whey or from buttermilk, the principle is the same. You'll see here on your left-hand side says feed tank. You put the butter, the buttermilk, or the whey, and you have a pump that goes through what we call the filtration module. So imagine that diagonal line on that little rectangle is the membrane that we use. It's very special. Now we're talking about the milk-fat globule membrane, and this is a ceramic membrane. It's a man-made membrane. What it is the filter for all practical purposes. So if you follow the flow directly out of the feed pump, it goes there, and the liquid will find that filter. And that filter is very special. The low molecular weight components are going to go through the filtrate stream. We call it the permeate or filtrate. And the rest is going to be concentrated, and it keeps being concentrated the more loops that we go through. And that is very important because according to some of the processes that we have, we can get rid of some of the protein, certainly the calcium and the lactose, and concentrate our milk-fat globule membrane. For those of you that want to see how efficient is this, I can tell you to make 1 kilogram of the milk-fat globule membrane ingredient, you need close to 200 kilograms of whey, of milk, or any other component. And as is the case, and this is a very interesting part of something that is happening in our research field right now, as any filter, it can get clogged, and it can, really hamper our economy and our efficiency of this process. It turns out that what clogs this membrane happens to be material very rich in membrane. So this slide is a clear example that one man's waste is another man's treasure. Dr. Damodar and Srinivasan in Wisconsin spend a lot of time trying to better prepare whey to isolate the whey proteins that we use so much these days in whey protein concentrates, whey protein isolates, because he identified that if you see those test tubes have like a little sandy residue in the bottom, and that's what was clogging the membrane. So he devised a way of getting rid of it. And when we analyze that, that is very rich material for the milk-fat globule membrane and the phospholipids that I've been talking to you about. So this is just an illustration that you can have this membrane system for the laboratory or pilot plant, which is what I have here at the Ohio State University, but it has been scaled up to industrial scale to produce whey protein isolate, and whey protein concentrates, which is, by the way, one of the major economic impacts that we have had in our dairy industry. We use that in many foods. We export it, and it's right now, I would say, a byproduct of cheese, but I think right now there's more value on these fractions than there are even in cheese. But let me just go back for a minute about the importance of the structure of these components that we have been describing. How do we isolate them from milk? But in milk, what do they do? Well, I'm going to show you a picture that a former student of mine, Sophie Gallier, very artfully did of the really droplet of milk, droplet of fat in milk. And with a fluorescent microscope, she showed that there's these black spots, which happen to be what physical chemists call liquid order domains of phospholipids. These are so organized, so well structured, that they don't allow the fluorescent pigment even to go in there, and that's why we see them as black spots. The lesson that we got from there is that it complicates our studies, because clearly the surface of the fat globules, for that matter, the surface of the epithelial cells producing milk, are not homogeneous. And that has deep implications in chemical reactions, biochemistry, and being very interested in processing, I can show you in this next picture that even mild thermal treatments, like on your left, is cooling it to 4 degrees, which is how we refrigerate milk. You can see how that surface looks very rough, very rugged. And if we heat it up to only, say, 45 degrees centigrade, which is, a very hot day in California, for example, the surface is going to take a complete different morphology, very smooth, and we see very well-defined domains of these little black spots. Furthermore, now that we are very interested on how the microbiome gets affected by our food, let me tell you, it is not just that we have the sugars or the oligosaccharides or not whatever that they're feeding. We have seen that the bacteria bind very specifically, depending on the kind of bacteria, to this surface. And what we know now is that the interaction is mutual. Not only the bacteria bind to the fat to cause some probably chemical, very important changes and digestion, but because of that interaction, the milk fat globule can also be thought as changing the composition of the bacteria, because the bacteria needs to secrete other enzymes in order to get food from the fat. When we thought about this, I'm showing you a chromatogram, liquid chromatography, of the comparison of the phospholipids from milk on the top and from soy, or for that matter, any other plant phospholipid components, which are called lecithin. Up here, you would see that, to me, most important, the sphingomyelin, that SN that you see, that peak, is completely absent where it should be if you compare them in soy. If you ask me, that stands to reason, because there's no real compelling law that says that a soybean needs to produce a component that is very good for brains or growth on the intestines, since the beans really are not designed to be eaten. And I've looked, and the soybean doesn't have two very important things. The soybean does not have a brain or a mammary gland, and hence they don't need to synthesize those components. So we know that these microdomains forming the outer leaflets of these membranes and they have an organization. What that means exactly is still up in the question, but it's very scientific question, but it's very interesting. And we start by describing it. So where is the sphingomyelin, where is the cholesterol? And it turns out that these two components, sphingomyelin and cholesterol, play a very important role in what I call the health of the membrane. Now, where does the glycosylated lipids, like the gangliosides, where are they located and what role there is what several of my students are now studying. But here's an observation. It turns out that if we change the proportions of the phospholipids, we don't get those little black spots. So that tells me that it's important. And very recently, Dr. Contarini and her group published a review in which they compare many publications of milks from, as you can see, buffalo, goat, ewe, human, donkey, mare, camel. And what is remarkable is that the ratios of phosphatidylethanolamine, phosphatidylcholine, and sphingomyelin, highlighted in yellow in the upper part, always more or less compose 1/3, 1/3, and 1/3 of all the phospholipids present in the milk of all of these mammals. And when we obey this rule in making model fat globules, they do present these microdomains that I was showing in the previous slide. So these proportions are very important for the normal function of the membrane. And just the lower part is the same phospholipids, and it pretty much doesn't matter what we do, cow butter serum or cream or buttermilk, our best efforts to disrupt that, these three families of phospholipids remain attached together. Now comes a personal view of what is the importance of this membrane and its structure. In 2006, the PNAS had a paper by Robnek in which he wanted to see how deep one of these proteins that are in the milk fat globule membrane could get. Did it get all the way to the lipid core? They're very elegantly shown here on this freeze fracture scanning electron micrograph. And you can see clearly that, yeah, there's no question there's three layers, a monolayer and then on top a bilayer. But what I want to call your attention is that he also published with no explanation these little bubbles that were present there. And really they're not in the size or morphology of the domains that I was showing you in the previous confocal microscope. So for many years we were wondering, what may that be? And it wasn't until 11 years later or so that my same student, Sophie Gallier, now working in New Zealand, she had to go all the way to New Zealand to work with a friend of mine, Dr. Harjinder Singh, and they did a study of this phospholipid membrane in vitro digestion. And the surprise to me was that you see there 180 minutes. This is a picture of a fat globule, normal in cream, and you can see those little kind of blisters on the surface of the fat, which denotes that the membrane has not been digested, but at the same time the triglycerides inside that sphere are being digested. And so that is really telling us, and this is for future studies and how we probably will design the formula or even the drinks of the future on how we really should digest the fats more slowly or in a very regulated way. So that's a lesson for all the nutritionists out there. I think that this is very important. More in the technological way, we really want to know what is happening to our powder ingredient. What do we do? And that powder ingredient, believe me, we're analyzing it in many ways. None the least is by something that is like the atomic force microscope. This is a laser three-dimensional microscope. And what you see there is a little droplet of fat, this is not, of powder, this is not at the same scale of what I was showing. This is just that minute granule of powder, and in your left-hand side is how it comes with a lot of the triglycerides in the surface, and then we've been tasting them with we've been processing them with some new moderately process, which called supercritical fluid extraction. You use a CO2 at very high pressure and temperature, well, not very high, a couple of atmospheres, and that makes it work as a solvent. But interestingly enough, it just dissolves the triglycerides, leaving intact some of the other proteins, and it is a process much milder than some of those thermal processes we make. In this slide, you will see on your left-hand side a very nice, when my student runs gels that look like that, I think they're ready to graduate. These are SDS protein gels, and in lane number 1, you have your molecular weight standards. Lane number 2 is perfectly preserved raw milk. Number 3 is refrigerated milk. And 4, 5, and 6 are increased heat intensity treatment of the cream that we use to prepare these proteins. Well, I'll draw your attention to the bottom of this, and you'll see as the heat treatment increases, the band that is showed at where it says beta-lactoglobulin height, which is, at 18,000 molecular weight band, you'll see that this low band that is shown 3 here increase. Yet in number 7 is what we get from the treatment with the supercritical, which doesn't seem to really even compare to a mild pasteurization process, as it's number 4. So that is a very hopeful thing that we probably are in the verge of developing very exciting technology that will preserve right now all our, hopefully, bioactivity as it comes from milk. Let me just change gears here a little bit and start talking about our evidence for the biological activity of these components that I've been describing. Now, a little while ago in 2011, with my friend Bob Ward, he designed this experiment, and we were just supplying the milk fat globule membrane material. So, if I draw your attention to the squares here on your left-hand side, so there were six mice that were just fed, 12, that were fed a normal diet. Some of them had saline injection, and 6 of them, we challenged them with lipopolysaccharide, which is a toxic component that causes inflammation and other kind of really toxic problems, especially if you have a subject that has a leaky gut. But knowing that that's what we wanted to do, we have exactly the same mice, same strain, and we gave them exactly the same feed, except we added the milk fat globule membrane. 6 of them, we gave them an injection of just saline to see if there was any response to that, and we challenged them just as the normal fed ones with lipopolysaccharide. The nutritionists in the group were looking at the consumption of feed and the body weight gain was no significant different. Where there were very important differences was, and I love the statistic of this experiment, the first 2 squares on the graph here shows what happened after 24 hours or 48 hours of the saline injection to the mice that were normal diets. And as you can see, well, they felt something, but there's no significant difference on their plasma fluorescence. However, the next square that you see, the plasma fluorescence of the normally fed mice, but challenged with LPS, went to the roof. And unfortunately, at 48 hours, all these mice, all of them, all 6 of them, were dead. Deader than a doornail. If you see the dark squares, whether these mice that were fed the milk-fat global membrane, whether they were injection of saline or LPS, there was absolutely no difference in their plasma fluorescence, and they all were alive and well. So that is a great result in my book. No need of statisticians or anything like that. But the reductionist mind of scientists wanting to see what is the one component that is doing this. And I have there involved, this is a complex material and makes it impossible to associate the stress protection with any one single component. I may even add, I think that we have to have the right composition, the right ratio of the components to achieve these results. Let me move to another example. The next one is how these components modify the tissue inside of the intestines of these mice. The body weight, the villus length, and everything were the same. And what we have is a group, the gray squares denote the mother's milk. That's pups, the little mice that were fed with the milk of the mother. And then the control is just that were fed similar to an infant formula, and the white blocks in the middle were 2 doses of the milk-fat global membrane. As you can see, the body weight, the villus length, it changed a little bit. But where it was dramatical difference is in this picture that I'm going to show you. So if you go to the left end of the squares, you can see the mother milk fed unchallenged villi of the intestine of these pups. And the 2 hours after the toxin, mother's milk gave perfect protection. There didn't seem to be much degeneration of the tissues. But in the control, which was the infant formula by itself, and it was mostly with soy components, you can see that there's a dramatic decrease on the villi of the intestinal lining of these animals. When they were fed exactly the same formula, but with added milk-fat global membrane, well, you can see, especially in the middle picture, that the tissue resembled much more the one of the tissue of the pups fed the mother's milk. Something like that was also observed when these same tissues were looked at the blue fluorescent, in which the intensity of the fluorescence on the mother's mouse was compared to the fluorescence of the control, and you can see that data on the bars all the way to your right, and you can see that the mother's milk and the formula with milk-fat global membrane are much more similar than those in the control. So that is very exciting, and I picked just this one paper to show something that I want to really stress the point on the biological activity. We all know that I have a daughter that is specialized in cognitive science. She's got a PhD. But all of the cognitive tests have to do with teaching something and then seeing how rapidly somebody teaches or not. And this paper by Thompson, published in 2016, really didn't have to look at that. It only looked at the non-REM sleep, which this REM sleep is just the normal response of our central nervous system to something as normal as sleep. And here is something very interesting that they did. They analyzed not only that, but they analyzed the microflora, the microbiome on these control mice and the test mice. And you can see that once they had the milk fat global membrane, the colony-forming units was enhanced a lot. Well, so was their microbiome. There were very interesting changes in the populations on the microbiome, as you can see there, and I'm not going to go into the detail, except for highlight the conclusion of these scientists, in which they showed us that the bioactive milk fractions, lactoferrin and milk fat global membrane, not only help promote the growth of beneficial gut microbial species, but they may affect the brain function directly. But that is something that I really need to understress for this audience. So effects that has happened, big industry. A Danone company is looking at the same pictures that my student Sophie Galliel, she worked for them for a little while, and they say, how can we humanize this? So there's been some efforts, and I'm going to go back to this slide in which you can see the domains there in your upper right-hand slide, and then you can see the distribution of proteins in the lower one on the right-hand side. And then by doing some process that they don't disclose, you can see that, this is bovine milk looking a lot like human, still not perfect, but it is very important. Now, rapidly, I want to move to the clinical trials, the study of TIMBI, in which they use 160 children trained on cognitive ability. All of this, you can read, decreased incidence of otitis media and everything, what results of this. Let me just show you very rapidly the result. And the very far end, on your right-hand side, mother's milk, response 106.4 on their scale. Formula fed, response only 101.8. But exactly same formula, but added with milk fat global membrane, those babies responded and their score was 105.8, very similar to mother's milk. And this was only in the cognitive test. The motor or verbal skills did not change, but for my pediatrician friends, there was a very significant reduction on otitis media, and those ear infections are really a plague for formula-fed babies, and that got reduced tremendously. Nothing to do with some non-invasive bacterial infections, which give to everybody. The significance of this finding was that I'll call your attention to the results of the meta-analysis of all of these papers, in which on your left-hand side were formula-fed benefits, and on your right-hand side, the breastfed benefits. Of course, there's not even touching the line at zero, which means that there's very significant difference between formula-fed benefit and breastfed. But if we were to increase, and we do with the addition of milk fat global membrane, that score all the way to 4, well, you see that the comparison would be a very, very important one. Now, you may be asking, well, this is fine and dandy, but how much do we need? And really, the amount of milk fat global membrane that we need to affect the change is not that drastic. Let me give you an example of what this was, a coincidental information and observation here at Ohio State. I have a colleague that is looking at adipocytes, and we saw that these adipocytes secrete membrane material that is very similar to the one on the milk fat globule. And what you see is the effect that these phospholipids have on a neuro cell culture. These are neurons in vitro cell culture. So the panel on your left-hand side is just with a wild-type cell, and the one on the right hand, in which we see a dramatic increase of cells, you see this growth, but these are only with the secretome of the adipocytes that are very similar to milk. very interesting that nature would have adipocytes that are having, milk-like phospholipids. So, of course, we wanted to link into that, but as luck would have it, I got bitten by some of my colleagues in Ireland, in which they published this paper, and I'm summarizing the results here. Panel A is the control neuronal cell growth. B is with the most advantageous, and cell culture know that 1% fetal calf serum promotes the myelinization and the growth of neurons, and you can see clearly how they are. But guess what? 75 micrograms per liter, or 150 micrograms, that should be a mu for micro, not an M, you can see a tremendous change on growth. And this is a direct effect that we have even in cell culture. So I think I have shown some, compelling evidence that as we process our dairy products, we can really modify the bioactivity of our infant formula. So I will leave you with some of the key points. The milk-fat globule membrane is a complex structure that contains bioactive fats and proteins naturally produced in breast milk. The human and bovine milk-fat globule are compositionally similar and share key bioactive components, and as we can see, they do have an effect nutritionally on brain and cognitive development. Now, the advances in processing technology are allowing us to isolate these very components from bovine milk so that we can add them into formula, and this formula is being shown that it functions closer to breast milk. I think the future is bright, and as Sun Tzu says, the opportunities multiply as they're sized, and I'm going to leave you with an image of me very happy working with my butter churn, and I think it's now time to see if I can answer some questions. Thank you very much.
Thank you, Dr. Jimenez Flores. So now we invite you to ask questions to of our faculty. If you have a question, please type it into the question field on your screen. Dr. Jimenez Flores will take as many questions as time allows. Our first question: is the milk-fat globule membrane structure derived from cow's milk similar to the milk-fat globule membrane structure in human milk?
Yes, the milk-fat globule components are one-to-one. Now, whether that is presented in the formula at exactly the same structure as with fresh milk, either from the cow or the mother, that's another issue. What we can draw is a perfect parallel on the composition and the ratio of the components that are there. We have proteins, as important as I was talking about, lactoferrin, mucins, and they all form integral part of the milk-fat globule membrane, which is formed by phospholipids, the phosphatidylcholine, ethanolamine, and the sphingomyelin. And those are very similar and very synthesized in the mammary gland in very similar ways in any of the animals and the mammals.
Thank you. Second question: is the bioactivity of milk-fat globule membrane derived from cow's milk unaffected during processing, and is there an assay to verify activity?
Okay, the second part first. Unfortunately, we don't have exactly a biological activity that tells us, well, this preparation really helps cell growth or neuronal growth or not. Hopefully, we're working towards that end. But we know that it's very important to have all of the components present. That means all the phospholipid components, including sphingomyelin, phosphatidylcholine, and phosphatidylethanolamine, in the same proportions. And that we can measure very accurately with techniques of HPLC, etc. As a matter of fact, I think that should be, if not is, because I'm not in the business, but it is what I would say is a perfect quality control for the ingredient that we add to the formula, is to make sure that all the components are there present and at the same proportions.
Thank you. Our next question is the bioactivity of the milk fat globule membrane derived from cow's milk and added to infant formula the same as the milk fat globule membrane bioactivity in human milk?
The answer to that question, I think I showed, is mostly yes. Since we can recover effects in the intestine that you can see using mother's milk as the control, our golden standard, and then comparing it to a formula without the milk fat globule membrane, the comparison were always very bad. Anyway. And the moment that we added these components in, a small concentration, we don't need it, tremendous amount, but we need to have it in a significant amount there, and milligrams per liter, we saw the effect. I mean, there was an effect not only on the cognitive thing, we saw the effect on the tissue development, on the POPs. We have seen the effect on the infection, the otitis media, for example, although that experiment also included something that is present in the milk fat globule membrane, protein called lactoferrin. So you may split hairs and say, well, what would be the effect just with lactoferrin? And while I don't doubt that there would be an effect, probably the measurable, the important thing is that there is a synergistic effect when that lactoferrin is in the realm on the contextus of the milk fat globule membrane components.
Thank you. How much milk fat globule membrane is in formula, and how does this amount compare to the literature?
Well, the amount that is being added to the formula is such that it mirrors and reflects the amount that was used in the review of the experiments. I don't want to say mine experiment, but the ones that have been talked and the ones that have been studied. So there is also an important part that beyond a certain amount, I'm going to change here the slide, in which we have these there 1.2 grams per liter or 6 grams per liter on the preparation that has a fraction of those 1.2 grams is the phospholipid. But as you can see, there was not that big of a difference going from 1.2 to 6. Same thing that we saw for the neuronal development, that while there is 75 micrograms per liter or 150 micrograms per liter, there's one data missing there, that when they went to 300 micrograms per liter, there was no further increase or change. So how are we going to dosify that is, first we're going to see what was done on these experimental trials and mimic that amount into the infant formula.
And our next question, how much phospholipid is in milk fat globule membrane?
I would say that if you isolate just the membrane, it represents about 60%. The other 40 to 45% are the proteins present there. The membrane material is almost exclusive phospholipid and protein. Now, as I showed in my picture, if you isolate the cream, the amount of phospholipids in cream only represent, say, 0.1%, maybe 0.2% of the total lipid present there. That's why it's important that the technology that we develop can concentrate this component to transfer it from cow's milk into our infant formula.
Thank you. Our last question, what is the most efficient way to derive milk-fat globule membrane from bovine milk at this time? Then it follows with through buttermilk or whey.
Right now, because of all the advances that we have had with whey processing, I have to say that the ingredient that is most likely to be utilized immediately is from whey. And it obeys to also that the milk used in cheese making is really not thermally abused that much, because everybody knows if we overheat our milk to make cheese, we don't end up with a good cheese. So we like the fact that it is going to be treated nicely and reduce all risk of microbial contamination, but it's also at the same time not thermally abused. And I think that's going to be the first commercial way. The second one will be buttermilk once we get over the hurdle of the thermal treatment to cream for butter, because it's certainly much more abundant in buttermilk than it is on the whey fractions that have been used up to now.
Wonderful, thank you. Our deep thanks to Dr. Jimenez Flores and to you for joining us. Slides for today's webcast may be downloaded under the Additional Resources tab. So I want to thank Dr. Jimenez Flores again, and on behalf of the Annenberg Center for Health Sciences, we thank you for your interest in applying milk-fat globule membrane and human milk science to improve nutrition for infants and toddlers. This ends today's program.